Okay, this lesson is to demonstrate how to use the SPICES format in our ninth grade living environmental class. This template is created to help you figure out the appropriate way to design an inquiry-based lab based on a question given. So, for example, SPICES stands for situation, prediction, investigation, collection of data, evaluation, and steps for the future. It's the same as problem, hypothesis, experiment, observation, and conclusion. All right, so just to go through it to show how it works, first you'll be given a question. You could choose from one of these formats that's here. You could say the purpose of this experiment is to, or our favorite one to use in class is does blank affect blank. So that would be our question. This question will be given in class, or depending on the setup, the student may decide on figure out the question themselves. So after the question is determined, then the student has to come up with the prediction, what the hypothesis would be. Here are two formats that the student can use to create the hypothesis. Our favorite to use in class is blank will influence blank. There's a connection to using the blanks. The blanks on the second dots represent the first dot represent the independent variable, and the second dot represent the dependent variable. So once we've filled those in, we could move on to the investigation, which includes our materials, procedures, step-by-step procedure. And in the setup of the procedure, you will need to create a control group and experimental group. The control group is the group that is used for comparison. It makes the experiment valid and reliable. So you're going to compare your data to that group. The experimental group is the group that we're going to use to test our hypothesis. And it usually contains one variable that's changed. And so this way we can maintain it being a controlled experiment. Within this section, investigation, students will identify what the independent variable or the dependent variable is. And it's very easy to go back to the original question and pick out the independent and dependent variable, or from our hypothesis to figure out what the independent and dependent variable is. Another variable is referred to as our control, or we mostly call it our constant variable. And these are the things that will stay the same in both the experimental and the control group. So all of these things must be identified in the investigation portion. After that's completed, then students will need to create a data table. The data table should reflect the variables on the data table itself. So if you were to create a T-chart, the T-chart, the x-axis would represent the independent variable, and the y-axis would represent the dependent variable. That will give you an idea of where to put the data you're collecting. The analysis portion is basically the conclusion of the experiment. It should support your hypothesis and answer the question. But in this section, it's very important that data is used to back up the information that you're concluding. So you should refer back to the data table with the numbers or what you have observed happening to back up your conclusion. Steps for the future is the last step, and this includes areas where you list steps for improving the experiment. You could list variables that may have influenced the outcome, or discuss source of error that can affect the reliability of the experiment. So that's pretty much SPICES. Every now and then we'll take it a step further, where I'll ask the students to come up with 2 suggestions on how the data that you have collected from this experiment can be used to create another question. So this is where we'll take the experiment to another level. So this is a template that's usually given to the student, and they will just pretty much fill it in themselves. This is in the beginning, but as we move on throughout the year, the students are then encouraged to create this on their own. And so here's an example of one that we did in class. The question was, does the concentration of the solution affects the size of the cell through osmosis? And so once again, you can see here the purple represent our independent variable, the blue represent our dependent variable. And so it's just a matter of transferring it down. And this is just to get the ninth graders used to identifying these variables from the beginning of the experiment. So here's the setup. So the control group, we had the isotonic solution. Here we show that the variable was the different solutions that they were put in. So we had the hypertonic and hypo and hypertonic solution. The independent variable represented the concentration, the different concentration of the solution we had, and what we were looking to see, which was our dependent variable, the response we were looking to see is the change in the size of the egg. Some of the things that stayed the same was the container, the number of days we had, and the egg itself remained as our constant variable. So you would then create a T-chart to represent this data, and then you could answer the question. Some of the most common things to write for improvement would be to add more trials, to repeat, to add more samples, and so forth. So these were just some of the things we came up with in class. So once again, this is SPICES, the acronym. I did not create, I found it online somewhere, but the details that went into creating this, I based it off the Regents' requirement for setting up an inquiry-based lab. So that's it. SPICES format for ninth graders to help them create an inquiry-based lab. Thank you.